Hello and welcome to Nikolay's genetics lessons. Yesterday I got a problem from one of my subscribers, and so this is very easy problem to solve. Still, I believe that many people would have a problem to find a solution, and today's video going to be probably long because I'm going to talk about many different conceptual ideas in this video. But still, I recommend you to watch it because this video was probably 10 short videos. So you would be able to solve many different genetics problems after you watch this video. A pure plant with round seeds having large starch grains is crossed with another pure pea plant with ring seeds having small starch grains. The F1 heterozygous formed are self-pollinated. What are the phenotypic ratio of plants with round seeds and intermediate starch grains to plants with ring seeds and large starch grains expected in F2 generation? Please explain this problem. So I sent a message to this person that with information given it is impossible to solve this problem because it's not stated which trait is dominant and which recessive or phenotype of the F1 generation is not given. So I got another comment from this person, and this is this comment that I find rather interesting because it contains mistakes and mistaken assumptions, and I believe that many people would make the same mistake. So I want to show you this comment, and later I will explain what is wrong here, what assumptions wrong here. So I think that this is a very good way to explain something, not just to show the correct way to solve the problem, but also to show common mistakes that people can make. So here, this comment: in this problem, round seeds mean capital R, and large starch grains means capital L, or these traits are dominant. If this dominant, then it is crossed with ring seeds, means small r, and small it is emitted starch grains, means L, small l. In F1 generation we get 4 phenotypes, and here is a list of 4 phenotypes: capital R and capital L, small r and capital L, capital R and small l, and small r, small l. But they ask us the ratio of the phenotypes of plants with round seeds and intermediate starch grains to plants with ring seeds and larger starch grains in F2 generation. The correct answer is 1 to 6. So you also might try to solve this problem on your own, so all the necessary information is given, and I recommend you to pause video here, and later you can run video again and you can compare your answers with my answers and explanations. So I will proceed, and I will start with common mistakes, and the first sentence contains such mistakes: a pure plant, and there is no such things as pure or dirty plants. In genetics we call such plants pure breed plants or pure line plants. So what does it basically mean? That means if we cross one plant with some trait with another plant with the same trait, we always would get in the following generations plants with the same trait. There are not going to be segregation, or no new traits would appear in all the following generations. And we can get such plants if plants would be homozygous in the loci that code for this trait. For example, if we would have a plant with red flowers, so let red flowers or red color be dominant trait, so genotype would be capital A, and if we cross with plant that is going to be of the same genotype, as you understand, all the following generations would have capital A, capital A. So there is no any other variance here. So red flowered plant would always produce red flowered plants. And another situation, if we have a plant that is homozygous also for this trait but have white flowers, for example, small a would stand for the white color, and if we cross with another plant that is also homozygous for this trait, all the following generation would have flowers that is going to be white. But if we cross, as you see, for example, flower that is heterozygous, so capital A and small a, with another plant that is also going to be heterozygous, that is capital A, small a, as you remember, in simple Mendelian genetics, this still going to be red flowered plant. So if we cross a plant that is going to be phenotypically red with another plant that is going to be phenotypically red, in the following generation, we are going to get segregation of this trait. So here we would have capital A, capital A, small a here, capital A, small a here, and small a here. So as you see, one of the plants going to be with white flowers, and 3/4 going to have flowers that is going to be red. So once again, parental generation had only red flowers, and F1 generation, as you see, would have two traits. So one trait would appear that was not seen in the parental generation. So we cannot call such plants pure breed, because pure breed plants always would produce the same trait. So flowers that is going to be red here, and flowers that is going to be white here. So is it possible to find if a plant, for example, pure breed or not? Because this plant may have red flowers, but this heterozygous plant that is not pure breed also would have red flowers. How can we find it? And we can find it if we do a test cross. In the test cross, we are going to use plant of unknown genotype but known phenotype. In our example, this is going to be red flowered plant that can belong to the two genotypes. One can be capital A, another genotype can be capital A and small a. So we have to find whether this plant is this genotype or this genotype, whether it is pure breed or if it is heterozygous for this trait. And we are using another plant that we always know the genotype, and we always know the genotype of the pure breed white, because only this genotype can produce this color. So we call this test cross, when we cross unknown genotype with known genotype that is homozygous recessive, and we know this genotype. So this is going to be small a, and if we cross these two plants, we are going to get following genotypes: capital A and small a here, capital A, small a here, capital A, small a here, capital A, small a here. So as you see, in simple Mendelian genetics, this means that 100% result of such cross would be plants with red flowers. That means that unknown genotype is homozygous dominant, and this is going to be pure breed. But if we cross heterozygous plant with homozygous recessive, once again we call this test breeding, and this is going to be following genotypes here: capital A, small a here, capital A, small a here, small a here, and small a here. So as you see, 50% of such a cross would be plants that is going to be with red flowers, and 50% result of such a cross would be plants that is going to have white flowers. So that means that this plant heterozygous and not true breeding, or not pure breed. Let's now return to our problem, and our problem states that here we have two pure breeding plants, one with round seeds having large starch grains, another wrinkled with wrinkled seeds and small starch grains. And now this information tell us that here we have following genotypes. So one plant would be true breeding for the round seeds, and this is dominant trait, so we use capital R, capital R. This is going to be true breeding genotype, and we also know that this plant has large starch grains, and this is also dominant trait, so genotype would be capital L and capital L. Once again, it cannot be capital L and small l, because we were told that this is true breeding plant. So this plant have to be homozygous for both traits, for both loci in each trait, and another plant is homozygous recessive for both loci. So genotype is small r, and small l, small l. So it has ringed surface of the seeds and small starch grains. And when we cross these two genotypes, we are going to get F1 generation. So F1 generation, and this is parental generation, that is going to be heterozygous in all loci. So this parent on the left can produce only capital R. There is no other choice. This parent has only two capital R's, so capital R, and this parent only can give to the following generation only small r. So genotype going to be for this trait capital R and small r, and for another trait it's going to have capital L from this parent and small l from this parent. So this is going to be a genotype of F1 generation. And if we return to the comments of this person who sent me this problem, this person suggests that in F1 generation this is going to be the genotypes, or he called it phenotypes. As you see, this person makes a mistake suggesting that F1 generation would have only one allele for each trait. So this is genotype of the haploid organism, not of the diploid organisms. How this person made such a mistake, instead of using this picture that I draw here, his suggestion was that genotype of one parent was capital R and capital L, and we cross with another parent that is small r and small l. And now he starts thinking what kind of phenotypes these two parents can produce. Once again, we have two true breeding parents, and these parents are diploid. So as you see, this person suggests that each parent has only one allele for each trait, but actually each parent has two alleles for each trait, and then he mistakenly starts to mess up everything. That he starts to think, or she starts to think, what different combinations these four letters can produce. So this is the wrong way how to solve this problem. As you see now, 100% of the F1 generations going to be of this genotype. So how we are going to find all the possible variants in the following F2 generation? According to our problem, we were told that F1 generation were self-pollinated. That means this genotype were crossed with the same genotype. In other words, capital R, small r with capital L and small l. And 100% of F1 generation, as you see, belongs to this genotype. If we self-pollinated it, that means we have to cross this genotype with the same genotype. So, what is the possible variance of the F2 generation? This is once again very easy to find. Let's find how many different gametes this genotype can produce. And parent 1 and parent 2, as you see, have the same genotype. So we don't have to find genotypes for each parent. It would be enough to find only for one of the parents. So the first type of gametes would be combination of capital R and capital L. The second combination can be capital R and small l. So capital R and small l. And the third combination can be small r and capital L. So small r and capital L. And the last combination can be small r and small l. So these four possible types of gametes that this diploid organism can produce. So diploid organism would have 4 alleles, and haploid gametes would have only 2 alleles, but in different combinations, as you see. So if this is going to be male parent, for example, this would be 4 variants of haploid sperm, and if this is going to be female, this would be 4 variants of the female gametes that we call egg cells. In plants, of course, we don't call this sperm, we call this pollen grains. So now we see that each of these parents in F1 generation that we cross among each other may produce these 4 variants of the gametes. So let's now find what variants we may have in F2 generation. So one parent may have capital R and capital L. Another variant of the gamete would be capital R and small l and capital L, and small r and small l. So this is going to be parent 1, different variants of the gametes, and on this side we may have another 4 variants for parent 2. Once again, this can be capital R, capital L, capital R and small l and capital L, and small r, small l. Now we just have to build a Punnett square that is going to be 4 by 4 cells, so 16 different variants of the different genotypes. And now we can list all the possible genotypes in the F2 generation. So this is going to be F2 generation. So here we would have capital R, capital R, capital L and capital L, capital R and capital R here, capital L and small l here, capital R, small r, capital L here, capital R, small r and capital L, small l here, capital R here, and capital L, small l here, capital R here, and small l here, capital R, small r here, capital L, small l here, capital R and small r here, and small l here. As you see, all the F2 generation, of course, going to be also deployed just like parental generation. And this is going to be 4 possible variants of the gametes. And gametes, of course, would be haploid. So, capital R, small r here, and capital L, capital L. Capital R, small r here, and capital L, small l here. Small r here, capital L here. And small r here, and capital L, small l here. And the last column, capital R, small r, capital L, small l, capital R, small r, and small l, small l. Small r here, capital L and small l here, and small r, small l here. So this is going to be all the possible genotypes in next generation. Let's now find what is the possible phenotypes if we would have here simple Mendelian genetics traits. The first variant would be plants with round seeds and large starch grains. So this would be one variant. This would be the second. Those, as you see, we have heterozygous trait here, but still in simple Mendelian genetics, if we have dominant allele here and recessive allele, the trait still would be dominant. So this plant would produce large starch grains. Next, once again, the same phenotype here, same phenotype here, and next one, as you see, we have small l, so round seeds with small starch grains. So next one would be the same that we are looking for, and the same here, but one more here, but all the rest would produce different phenotypes. Let's find the phenotypes also. So here we would have different phenotype. There's going to be plant with round seeds and small starch grains. The same phenotype here, and one more here. And the third variant would be ringed seeds and large starch grains. and here is 3 variants here. And the last one would be ringed seed coat and small starch grains. As you see, we have here following phenotypes 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 to next variant would be 1, 2, 3. So 9 to 3 to next ratio would be seeds with ring seed coat and large starch grain, and we have 1, 2, 3 such variants. So 9 to 3 to 3, and we also have here one variant with ring seed coat and small starch grains. So we have 9, 3, 3 to 1 ratio here. So total number of the cells 16, and 9 plus 3 plus 3 plus 1 also would give us 16. Once again, this is going to be ratios if all the traits would be under the control of the simple Mendelian genetics rules. But as you remember, the answer given were that the correct answer is 1 to 6. So how is it possible? And if you take a closer look to this problem, you would see the answer. So the question was, what is the phenotypic ratio of plants with round seeds and intermediate starch grains to plants with ring seeds and large starch grains? First of all, in simple Mendelian genetics, we don't have intermediate phenotypes. As you see, if we cross, for example, two parents, these heterozygous plants would look like a dominant plant. But because we have a word intermediate starch grains here, I want to underline this intermediate. That means that this is not a simple Mendelian genetics, and this trait under the control of different rules. So I will explain here. So the first variant can be, I would use once again plants with different colored flowers. For example, if we would have one plant that is going to produce red flowers, and when we cross with another plant that produce white flowers, for example, genotype of this plant can be capital A, and genotype of plants that produce white flowers can be small a, and when we build the Punnett square, you would see that genotype would be capital A and small a, capital A and small a, capital A and small a in 100% of the progeny. This is going to be plant that produce red flowers, and this is going to be plant that produce white flowers. But for example, if this is going to be incomplete dominance, 100% of the progeny would have plants with intermediate color of the flowers. So F1 generation would have all the plants with pink flowers. So it would be different from parent 1 and parent 2, and going to be intermediate color between white and red. This is one possible explanation. Another possible explanation we call additive trait. For example, if one plant is, for example, 1 meter and we cross with another plant that is 50 centimeters, all the progeny going to be 75 centimeters. We call this additive trait. Once again, for example, genotype of this parent would be capital A, and genotype of this parent would be small a, and genotype of the progeny going to be capital A and small a. In this case of this parent, we can say that each allele here adds 50 centimeters. So 50 centimeters plus 50 centimeters would give us 1 meter plants. And here each allele would give 25 centimeters. So 25 centimeters plus 25 centimeters would give us plants that is 50 centimeters tall. And here we have one allele that give 50 centimeters to the tallness of the plant, and another allele that give 25 centimeters. So total tallness of this plant would be 75 centimeters. So as you see, F1 generation in this case also would be intermediate between two parents. So one parent 50 centimeters, another 1 meter or 100 centimeters, but the progeny would be intermediate, would be 75 centimeters. So once again, we call this additive trait, and this variant we call incomplete dominance. And what do we have in our example? We were told that one of the parents in a parental generation is homozygous dominant for this trait and has large starch grains. Another parent is homozygous recessive and has small starch grains. And when we cross these two parents, all the F1 generation going to be heterozygous for this trait. But genotype going to be different from parent 1 and different from the parent 2. So in F1 generation we got 100% of the plants with intermediate size starch grains. So for example, if this parent produce this size, this parent produce this size grains, so here we can expect something in the middle. So all these genotypes would produce intermediate size starch grains. So three possible genotypes, that is capital L, capital L, capital L, small l, and small l, would produce three different phenotypes. Large starch grains here, medium starch grains here, and small starch grains here. And here, in this example, genotype means phenotype. So we have three genotypes here and three phenotypes here. But in simple Mendelian genetics, we would have only two phenotypes here. So this phenotype would produce the same results as this phenotype. But whether this is additive trait or if it is incomplete dominance, in such examples, three genotypes would produce three phenotypes. Now we can return to our table that we built before, and let's now analyze this table in the light of this information. So now let's analyze how many phenotypes we would have here. So this is going to be one phenotype, but this is going to be different phenotype. This is going to be intermediate size starch grains. So we have to use different color here. Let me change the thickness of the line. So different variant here, and different also phenotype here, different phenotype here. So whenever we see capital L and small l, this means new phenotype, and different phenotype here, different phenotype here, and different phenotype here. So this phenotype would have round seeds and intermediate starch grains. Let's now return to our question one more time. So the question was, what is the phenotypic ratio of plants with round seeds and intermediate starch grains to plants with ring seeds and large starch grains? So let's now count what is the ratio, and we have 1, 2, 3, 4, 5, 6. So we have new ratio 6 plants with round seeds and intermediate starch grains. And let's now find how many we have plants with ring seeds and large starch grains. And I see only variant here. So small r would produce ring surface of the seeds, and capital L would produce large starch grains. So as you see, now we have our ratio 6 to 1. So once again, for example, here we would have ring surface of the seeds but intermediate starch grains. Here is also ring surface of the seeds and intermediate. So only one variant when we have ring surface of the seeds and large starch grains. And this is exactly the answer that person sent problems today suggested as correct answer that was given on the exam. So hopefully all this information will clear to you, and now you have ability better to understand and solve analogous problems. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.